Hi students, my name is Roy Lee. I'm here to explain what General Science Biology class is all about. So, in order to understand what we're learning, we have to talk about how the science curriculum is taught in schools. We start off with something called general science. General science is basically all the sciences combined together. So you learn about a little bit of biology, a little bit of chemistry, a little bit of physics. Now, once you get that stage over with, you move on to the next course, which is usually around 9th or 10th grade, where you start taking intro bio, intro chem, or intro physics. After those classes are done, you move on to more advanced placement courses, like AP Bio or IB Bio, where you learn about more advanced biological concepts, chemistry, and physics likewise. And then you move on to college level courses. So, who is this class designed for? This is for students that are in 7th to 10th grade. So, relatively younger students who are currently taking a general science class or intro bio class at school. And here are some common questions that students often have. Teacher, I'm worried about moving to another country. These are the students that are a little bit concerned about moving to an English-speaking country where they're going to go to an English-speaking school, where they're going to be taught science courses by an English-speaking teacher. Now, these are students that might have been quite advanced in terms of their science skills when they were in Korea, but because of the language barrier, they feel like they can't catch up. This class is designed for students like you. We're going to start with the very basics of science and explain everything in terms of English terminology so that you don't have to make that language barrier jump by yourself. Number two, there are students that say, I'm completely lost. These are students that have no idea what's going on. That's okay. This class is designed for you. We're going to start at the very beginning, the very foundation of all sciences, and try to build up our knowledge step by step. Number three, there are students that say, I want to start from the very beginning. I completely relate to students like this. This is how I learn things. I start from the very beginning, and I have to go at my own pace, slow and steady. If I start from the middle, I'd rather not start at all. This class, again, is a foundational class. It starts from the very beginning. So if you want to learn about biology, but you feel like you know nothing about biology, and you want to start from the very beginning, this is it. Finally, there are students that say, I don't like science. I have a few things to say about this. First of all, I don't really agree that you like or dislike science at this current age. Assuming that you're a student that are in 7th or 10th grade, and you say you don't like science, and I ask you, when did you first learn science? Students often say, around grade 5 or 6. So it's been about 2 years since you actually started an actual science course. You can't decide whether you like something or not just based on 2 years of schoolwork. If you go to college level and you major in a science and you say, oh, I think science is too hard for me, I completely understand that. But at the high school level, high school science doesn't really take a lot of, let's say, personal brain power. It can all be overcome by the amount of effort that you put in. And I know that I sound like an old Korean ajashi when I say this, but it's true. Science level courses, high school level science courses, can be overcome with effort. Whether you like or dislike sciences, it's irrelevant. So I understand that you might have preferences. Hold on to those preferences because those preferences build prejudices. And you feel like, because you don't like science, I'm not good at science. No, let's break that connection right now. Let's try to think about what science is, and let's learn about science at a very foundational level. Later on, we can decide whether we like science or not, okay? So this is a question for another day. So there are some recurring themes in bio. So what are we actually learning here? We're learning about biochemistry. We're learning about cell basics, cell respiration. We're learning about photosynthesis and genetics and evolution and ecology and physiology. These are all things that we're going to be covering. You'll notice something strange though. If you go to more advanced high school level courses, these are the things that you learn in biology. Notice something? It's the same thing. Biology is recurring themes. It's the same things over and over again, but each time you take a more advanced class, you're learning more advanced concepts related to the same themes. If you get to college level, it's the same thing all over again. You'll still learn about evolution. You'll still learn about cell respiration. Even if you go to medical school, you still learn the same thing. You learn about cell respiration, and you learn about the kidney function. So what I want to say is, if you're going to be studying biology for the next, let's say, 6 to 7 years until you get to college, you'll have these recurring themes that are going to come up over and over and over again. Now, imagine how a strong foundation will actually help you understand these future courses later on. this is the time to do that. We're building a strong foundation. We're learning about these different themes and the key concepts related to that. So that later on, when you take another more advanced biology course, the additional content is just building up on these strong foundations you already built up with me. So, what can you expect from this class? Better explanations than your science teacher. This is something that I can guarantee. If you have a school teacher that doesn't really teach you very well, or if you have a school teacher that just gives you packets that you have to fill out and doesn't really tell you about what you're learning, I can assure you that I'll have better explanations than your school teacher in the language of your choice. So I'm speaking in English right now, but I'm also fluent in Korean. So if you feel like you'll be more comfortable taking a Korean class, you have that option as well. I can assure you that you get a very good grade in general science biology because these are the themes that we're going to be talking about in this class. And because general science is not just biology, it's also chemistry and physics, I can assure that you get a good grade in this general science class overall. Finally, I want to emphasize again that what we're doing is we're building a foundation. Okay? And these recurring themes in biology is going to come up again and again. Later on in high school, later on when you take AP tests, later on when you get to college level, it's the same themes over and over again. So what we're going to do now is we're going to build a strong foundation as an investment for your future. My name is Roy Lee. I'll see you in class. Hi, nice to meet you. We're going to be studying general science together, and today we're going to be talking about the subject of biology. Like, as you know, general science is composed of 3 different scientific subjects, first being physics, second being chemistry, and third being biology. That's what we're going to be studying together. So since today is the first day of studying biology, I figured we would introduce the topic of biology together. We would find out what biology actually means. And based on that, we can learn about the different fundamental laws that govern all living things. And this is called cell theory. Now, biology, what is it? Well, it's the study of living things. Well, what does that actually mean? Well, the word biology actually has a very deep connotation. It has deep meaning lying inside the word itself. Bio is the Latin prefix for life. Think about different words that have the word bio in it. For example, antibiotics or biography. These are all words that are somehow connected with the meaning of life. Autobiography, for example, is a book that someone writes about his or her own life, right? So auto means self, bio means life, and graphy comes from like the word graphic. It means to depict something, to draw something. So if you combine that, it becomes the self-study of one's life and you draw out that life. That is what autobiography means. So bio, in essence, means life. What is logy then? I'm sure you've all heard of this word before, right? Biology. Logy comes from the Latin suffix of logic, right? What is logic? Well, when you try to solve a problem, for example, you have to use logical conclusions. You have to deduce logic from certain situations to solve that problem, right? So if you combine these two things, if you have bio and logic, it means to study living things in a logical manner. And that's what biology means. Okay? The thing you have to know is that it's the study of living things. And living things encompasses basically everything that you can think of. All life forms on Earth are living, right? So you have to study everything. Now, I want you to entertain me for a minute. I know you're listening, I know you're watching this video, okay? So I want you to close your eyes for a minute and do this thought experiment with me together, okay? So close your eyes and just listen. Now, here's what I want you to do. I want you to try to imagine a man, okay? Imagine just a typical man. Now, pretend that you are a camera director. You're some kind of movie director, and you're behind the camera, and you're adjusting the different zoom levels of the camera, okay? So you have this man in your mind's eye, and you're slowly zooming in on this man. Are you following me so far? Okay, so you're zooming in, and you get closer and closer. I want you to start zooming in on his face, okay? So zoom in closer and closer. You get to his eye. I want you to zoom into his eye. And then I want you to zoom in into his eye, okay? Zoom in even further. And I know you've watched a lot of movies, right? You've watched a bunch of superhero movies, like Marvel movies, for example. They often have this visual where you start zooming into like the cell, right? I want you to basically do the same thing. I want you to zoom into this eye, zoom in further and further. What do you see? You see globs of water and cellular masses. You see blood veins and neurons, right? And you zoom in and you come to one cell. Zoom into that one cell, okay? We're zooming into that one cell. Inside the cell, you see these weird pathways and you have this weird blob in the center that's called the nucleus. Zoom into that nucleus and you start seeing DNA. You know DNA with the double helix and like the little ladders, right? I want you to zoom into that DNA. I want you to zoom into the individual chemical molecules that make up that DNA. Are you following me so far? Okay, stop. Okay? That is the smallest unit of study for biology. We will not go smaller than that because if you go smaller, then it's no longer considered living. You're not studying a living thing. Then you're venturing onto the field of chemistry, okay? So right there on that border of understanding DNA, that's the smallest unit of biology. Okay, so close your eyes, we're going to do this again. I want you to, instead of zooming in now, I want you to start zooming out from that DNA, okay? So start zooming out, and you see the nucleus again, and you zoom out even further. Now you see the entire cell, zoom out even further. You see these groups of cells together. You have an aqueous, a watery environment. You see blood vessels. You zoom out even further. You see the entire eyeball, zoom out even further. Now you're at the man's face, zoom out, and you're at the original spot. You're seeing that man, okay? I want you to zoom out from that, okay? I want you to start zooming out from that. And what do you see? Well, next to this man, there's a woman, okay? I want you to zoom out even further than that, and you see that they're actually inside of a room in a second-story building. And then in the first story, there's two kids, a boy and a girl that's playing, okay? So obviously they're a married couple and they have two kids. And you also see a cat and a dog inside the living room. So it's a typical family with two pets. I want you to zoom out even further. Now you see the entire house, okay? You see the entire house. Zoom out even further than that. Zoom out even further and you start seeing the neighborhood. You see the neighbor's house. You see the street in front of the house. You see cars zooming past. You see trees and mountains and rivers in the backyard, okay? And you start zooming out even further and you start seeing the entire district, the city in which this man lives. Zoom out even further than that. And now you start to see the entire countryside. You start to see the state, for example. And you zoom out even further. Now you see the entire country. Let's say South Korea, for example. You start seeing South Korea, the entire borders, the oceans of South Korea. Zoom out even further, you start seeing Japan and China and other neighboring countries. If you zoom out even further than that, now you start to see the entire Earth with continents in it, with Asia, with Africa, with Europe, right? And I want you to stop there. Stop there. That's the maximum unit of biology. That's the largest unit of biology that we'll ever study. We're studying the entire Earth because, like I said, we're studying all life forms on Earth. And by extension, we can study the Earth as a whole. Okay, so open your eyes. Right? We've conducted this very simple thought experiment. We've tried to zoom in and zoom out on different things. And the thing you have to know is that everything that you saw in between the minimum and the maximum units, everything inside is considered biology because they're all living things. And think about what this means. Think about the number of things that you have to study. Not only do you have to know the human body, but you also have to learn about cats and dogs, and you have to learn about the trees and the rivers and the weather. You have to learn about continents. You have to learn about the different biomes that the Earth has. And not only at a macroscopic level, if you go down to the microscopic, you have to learn about DNA, right? And you have to learn about cells and tissues and blood vessels, but not only in human beings, also in dolphins and cats and tigers. And not only just animals, you have to learn about plants. You have to think about the Amazon forest. You have to think about the single individual tree inside an Amazon forest. You have to study that because that is studying living things, and we want to study living things in biology. How do you do this? it's impossible. It's too much information. Nobody in their lifespan can ever study all living things on Earth individually. That's just an impossible task to do. So, does that mean that we give up? Is there no way around it? That's not actually true. Remember what I told you about biology is the combination of the word life and logic, right? So we're trying to study life. Instead of studying all life, how about we try to use logic? We approach this problem logically, right? And what that means is we analyze life by fundamental rules. I'll give you an example. If you want to, let's say, study dogs, and you spend 40 years of your life studying dogs, you're a dog master, okay? So you study dogs your entire life. You know everything about them. You know their environment, you know what food they eat, you know their behavior, everything that's there is to know about dogs, you know. So, for example, let's say you want to study another animal now, right? You want to study the wolf. Does that mean that you have to spend 40 years studying the wolf? Well, that's technically impossible because you're probably going to be dead, I'm sorry. But because you've studied dogs, you can derive things from the dogs and apply them to the wolf and see if these rules have some sort of connection. For example, what can we derive? What can we deduce from dogs? Well, we can deduce that they're animals with 4 legs. We can deduce that they have fur, and they have snouts that look like this, and they pant when they're hot, and they're very friendly towards human beings. You can deduce all these logical conclusions from the study of dogs. And then you can try to apply them to wolves and see what matches and what doesn't match, right? So obviously fur matches, 4-leggedness matches, all these different things match, but there are a few differences. Hello, welcome back. Today is the 4th class and we're going to be talking about eukaryotes. Remember what we were talking about before? In the first 3 classes actually, we talked about the basics of biology. We talked about what biology actually means, then we moved on to cell theory, and then we divided living things into 2 categories, prokaryotic cells and eukaryotic cells, right? And last time we talked about prokaryotic cells, specifically the organelles that we find inside of those prokaryotes, right? This time we're going to be talking about eukaryotic cells. However, what did I tell you about the number of organelles? Do you remember? Yes, prokaryotic cells have fewer numbers of organelles. Eukaryotic cells have many more, right? Compared to prokaryotic cells. And because there are so many organelles, we need to divide the organelles into about 2 or 3 parts so that it's easier for you to understand, easier for you to memorize, and remember their functions. Okay, so this is the first class. We're going to be talking about eukaryotic organelles, and this is part 1. So, quick recap. What are we talking about? What do we mean by organelles? I told you organelles means small organs, right? These are organs that are found inside of the cell. Look at this. This is a typical eukaryotic animal cell. How do you know it's an animal cell and not a plant cell? What tips you off? Well, number 1, it's round shaped, right? It's not the hexagon shape that you found in the picture previously, which means that it doesn't have a cell wall. It doesn't have a rigid structure that protects the cell and is loosely based on like a circular shape and it looks like an amoeba. Yes, this is indeed a eukaryotic animal cell. Let's look at the organelles inside of the cell. Look at how many things are going on. So many things that are labeled here, right? And we're gonna start with this, the nucleus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, as well as the Golgi apparatus. And we're going to one by one cover all the organelles that we find in most typical eukaryotic cells, okay? Quick recap, prokaryotic organelles. We learned 5 things. I'm sure that you can, rat these off right away when I ask you to. 5 things. First being the plasma membrane. What was the plasma membrane? Do you remember? The plasma membrane was basically a small, thin layer. It's a membrane that surrounds the entire cell, right? And what is that membrane made out of? Do you remember? It was called the phospholipids, right? It was a phospholipid. A phospholipid is basically composed of 2 parts, a phosphorus group as well as the lipid group, right? And you combine these things together. And they kind of look like this. I drew a circle with a P inside of it, which represents a phosphorus group. And I told you that there's 2 legs that are extended outside of it. And what are these legs? These legs are basically long chains of carbon atoms, remember? I told you, think of it in like a necklace that your mom wears. It's a pearl necklace, and each pearl represents a carbon atom. This is basically like a long necklace. It's long chains of carbon atoms stranded together, and those are what we call lipids, right? So we learned about the plasma membrane. The plasma membrane is basically a thin layer, a layer of phospholipids separating the inside of the cell from the outside, right? Just in case you don't remember, let me quickly go over it again. This is a phosphorus group, this is a lipid group, and you have these phospholipids lined up together like, BFFs, and they're all holding hands, and they all create this one big layer. Remember that there was an opposite layer underneath this, right? We had the lipid part in the middle, we had the phosphorus groups on the outside, and together these formed this really big membrane, and this membrane basically covered the entire cell, and that's what we meant by plasma membranes, right? Let's move on. I want us to focus on the last organelle that we learned about prokaryotes, and that was ribosomes. Do you remember what ribosomes were? Ribosomes were shaped like a bullet, if you remember, and they're divided into two subunits. There was a large subunit and there was a small subunit. And what was the function of the ribosomes? The function of the ribosomes were that they attached themselves to the big circular prokaryotic DNA, remember, right? And these ribosomes, as they moved across this circular DNA, they created something called proteins, right? So ribosomes, they create proteins based on the information from the DNA. Why are we doing this? Well, these were the 5 organelles we learned about the prokaryotic cell. The eukaryotic organelles are actually almost the same. Aside from these two things being the nucleoid and the plasmid. What were the nucleoid and the plasmid? Quickly. Right? The nucleoid was basically a region where the DNA was located inside of a prokaryotic cell. What was the plasmid? The plasmid was like a superpower, I told you, right? It was an independent DNA, independent circular DNA that's not used every day, but in really special circumstances, it can be very useful. That's why the cell keeps it inside of itself, right? Aside from these two things, these 3 things actually are eukaryotic organelles as well, meaning that you can find these organelles inside eukaryotic cells. I put a triangle next to cell wall for a reason that most students would probably catch immediately. Why did I put a triangle there? That's right, eukaryotic cells have 2 different forms, right? They have the animal and the plant cells, right? Do animal cells have cell walls? We just talked about it, right? Animal cells are more spherical in shape and they're not rigid. They're kind of like jelly. Therefore, they don't have a rigid structure that's protecting them. They don't have a cell wall. However, plant cells do. Plant cells do have cell walls. And both animals and plant cells combined together are both eukaryotic cells. So it's hard to say whether or not eukaryotic cells have cell walls or not. It depends on what type of cell you're talking about. However, overall, you can say, yes, you can find cell walls inside eukaryotic cells simply because of the existence of plant cells. Another thing, we talked about ribosomes last time. Do you remember something that I mentioned about ribosomes that you should keep in the back of your mind? I told you that ribosomes are 70S. Do you remember? Prokaryotic ribosomes were 70S, and I told you not to worry about what S means. S is just a unit of measurement. It's a unit of size. I told you to remember the number, 70. Well, the reason why I told you to remember it is because eukaryotic cells also have ribosomes, but they're slightly larger in size. One more time, prokaryotic ribosomes are 70S, but eukaryotic ribosomes are 80S, meaning that eukaryotic ribosomes are slightly larger in size. And this is actually very important later on in the field of medicine, actually. When you have antibiotics, for example, and you're trying to kill bacteria, but you're also trying not to kill your own cells, then you have to create medicines that are based on this difference in ribosomes. And more often than not, antibiotics that target prokaryotic ribosomes are very, very effective. And that's that's why we learn about this. In general science, maybe your teacher might skip over it, maybe your teacher doesn't mention it too much, but later on when you get to SAT 2 biology or AP biology, for example, this becomes really, really important. So it's good for you to know this right now. Prokaryotic ribosomes are 70S, eukaryotic ribosomes are 80S, meaning that eukaryotes are slightly bigger. So this time we're going to be talking about eukaryotic organelles, and there's a lot of them, I told you, right? We have 5 over here, and we also have an additional 5, so a combination of 10. There's actually a lot more than this, but this is just the basic organelles that you have to know. So there's 10 of these, and you have to know their name, you have to know their function, you have to know where they're located inside of the cell, and you also have to know what they look like. So there's a lot of things to remember. So I feel like it's best if we break it down into 2 parts, right? So today, we're just going to be talking about the first 5. Thankfully, among the first 5, these 2 we already did, right? Plasma membrane. I don't need to go over this again. Again, it's phospholipid membranes that surrounds the entire cell. And ribosomes, they are small bullet-like shaped organelles. And what do? They read the DNA and they create proteins based off of it. So today, we're just going to be covering these 3 organelles and then we'll be done. Sounds good? Okay, so let's start off with the rough endoplasmic reticulum. This is what it looks like. What does it look like to you when you look at this? First of all, where is it? Where is the rough endoplasmic reticulum? Well, let me start off here. This circle shape over here, this sphere, is actually the nucleus. What was the nucleus? Do you remember? The nucleus is basically a container that contains DNA. And that container itself is also made out of membranes. Membranes meaning like thin layers of skin. Not your actual skin, but skin-like structure. So this nucleus has a nuclear membrane, nuclear envelope. So this is basically a membrane that creates this nucleus that contains all the DNA inside. And the membrane itself is actually extending outwards. So this entire thing is connected to the nuclear membrane. It's all one membrane. There's a sphere in the middle, and this membrane extends outwards like this. Hi, welcome back. Today is the 7th class, and we're going to be talking about cell respiration. Now, what have we learned so far? We started with the basics of biology. We've moved on to cell theory. Then we talked about prokaryotic, eukaryotic cells. Then we talked about the different organelles inside of the cells, right? Now, starting from today, this is where it starts to get a little sciencey. It gets a little sciencey. And what I mean by that is we start to talk about concepts that are not just memorization. Everything you learned previously is simply like, oh, I get it, right? I see how they categorize things. I see what the names of the organelles are. I can kind of memorize the functions, right? Starting from this section, we need to think about things that happen in the universe in a conceptual, in a scientific state of mind. And you'll see what I mean when we begin to talk about ATP and energy, right? So today, the topics we're going to be covering is ATP, and secondly, we're going to be talking about glycolysis. Okay, so let's move on. So energy. Let's talk about energy for a second. What is energy? When do we use the word energy? Well, we often use the word energy when, let's say, you need to do something. For example, hey, can you run to the post office and run some errands? Often you say, nah. I don't have energy. I don't have the energy to do that. Or, I need a cup of coffee because I'm lacking in energy today, right? So we use the word energy extensively, but what is energy? Have you ever thought about this? What is energy? Well, the thing you have to know about energy is that there are many different types of energy in the universe. And some types that we should know are some thermal energy, chemical energy, electric energy, and nuclear energy. And these are all different types of energy. But what is it? Energy, basically, think of it like this. Energy is something that you need when you want to do something. When you want to do something, what you need is energy. And this might seem just really intuitive, like, of course, obviously, right? But let's really start thinking about this in a scientific manner. Let's say your battery died on your cell phone. Okay, your iPhone died, it has no battery. What are you going to do? You plug it into the wall, right? You plug it in and you recharge the battery. What is actually happening here? Well, you're transferring energy from an outer source, from a power plant, and you're transferring it through wires into your iPhone. into the battery of your iPhone. Right? So later on, when you take it off, you have energy that's stored inside, and you need to do things with your phone, right? You need to call your friends, you need to text your mom that you're going to be late. All these things you need to do, and in order to do that, you need energy. The iPhone needs energy. Therefore, again, energy. What is it? It's something that you need when you want to do something. Okay. Let's talk about the different types of energy. What do I mean by thermal energy? Well, thermal energy is just a fancy word for heat energy. For example, if you're standing outside in the sun, what happens? You get uncomfortably warm. What is happening? Well, you're receiving thermal energy from the sun into your body. You're absorbing thermal energy. Make sense? Chemical energy. Chemical energy is basically, think of it this way. Everything is made up of atoms, right? All physical things in this universe is made out of atoms. And there are bonds that exist between these atoms, and we call them chemical bonds. For example, we talked about phospholipids, and we talked about long chains of carbons, right, in the lipid tail. And we said that these carbon atoms are connected together like pearl necklaces. So this one carbon atom is connected, it's bonded to another carbon atom, and there is energy that is stored inside of those bonds. And think of it like a spring, right? So two atoms, and they're packed tightly together, connected with the spring. What happens if I cut the spring? Right? The spring flies off like this. What is happening? Energy is being released. Chemical energy that was stored in the chemical bonds are being released to the outside world. And that's what I mean by chemical energy. What about electric energy? What is electric energy? Electric energy, it gets a little hard. And when we start talking extensively about electricity, then we move on to the realm of physics, right? And we're not studying physics right now, we're studying biology. So for all intents and purposes, let's understand electric energy to be simply a flow of electrons. A flow of electrons. And even that is a little too hard for biology. So think of it like this. Electric energy is basically what happens when you plug in your iPhone into the wall. Right? Some sort of energy that's generated flows through the wall into the wire into your phone, and your phone has stored electric energy inside of its battery. That's all you need to know about it. Nuclear energy, way too hard. We're not going to be covering nuclear energy today. But chemical energy is something that I really want you to think about extensively, because this is something that you probably haven't thought about before. Let's move on to the second point. Energy cannot be created or destroyed, only transformed into different types. This is actually a really important statement. This is called the first law of thermodynamics. And we'll probably talk about this when we get to physics. But for now, let's just understand that you can't create nor destroy energy. Energy simply exists, and what you can do is you can transform the energy into different types, but you can't just spontaneously generate energy. That's not possible in our universe. Okay? And how do you know? Well, just trust me on this, because there's people way smarter than you that are working on this, and they have proved over and over again that this rule is true. Okay? So let's think about this in an everyday example. Let's try to apply our scientific brain into something that we do every day. Let's say you want to cook some noodles. Okay? You want to cook some instant noodles, so what do? First thing, you get a pot, right? You get a pot, you turn on the tap water, you put in cold water, right? And you put it on the stove. And then what do? You turn the dial, it's like click, 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 and then fire comes out. Okay, what is actually happening here? Let's try to analyze what is happening here in a scientific process, okay? So you put in water, the water is cold, okay? You put it on top of the stove, and you turn the dial, right? And then suddenly these flames come up. What is happening here? Well, you understand that gas is flowing into that system, right? So gas that's produced somewhere else flows into your house through a hose, and it's open, it's released onto this gas stove. There's a little electric spark that begins a combustion reaction, which is basically the meeting of oxygen molecules with whatever molecule you're trying to have a reaction with, and it's a very, very violent reaction. Okay, so it's a combustion reaction, so you have a fire. What is happening? Well, how do we explain this in terms of energy? Well, gas molecules, right? The gas that's coming inside of your house is composed of gas molecules, right? And these gas molecules all have chemical bonds within these molecules, and they're very, very high energy chemical bonds. That's why they're so combustible. That's why they can light on fire. They can be lighted so easily. So there's a lot of energy stored in these gas molecules, and they're released. They meet with the oxygen in the atmosphere, with a little electric spark that generates that entire process. We have fire. What is fire? Fire is nothing more than light and heat energy together. right? So you are transforming the chemical energy stored inside the gas molecules, and you're converting it into heat and light form. Well, how do you know it's light? Well, you can see the fire, right? That means the light is produced, it goes into your retina, and you can see it. What is heat? Heat is thermal energy, right? So you generate heat, and the heat heats up the pot. The heated pot heats up the water, and then you have boiling water. What is boiling water? Right? And again, think about this in terms of energy, and it gives you a whole new perspective on what is actually happening here. What is boiling water? Well, we understand that this water is gaining energy, right? Hot water and cold water, which one has more energy? Well, obviously the hot water, right? How? Well, where is that energy going? I mean, where is it? Where is that energy? Think about it this way. When you put water in a pot, right? That water, if you go down to the microscopic level, that water is made up of water molecules, right? And we all know what the chemical formula for water is H2O. So there's 2 hydrogen atoms and 1 oxygen atom, and it looks like a Mickey Mouse form, and you've all seen this before, right? 1 big oxygen atom and 2 hydrogen atoms attached to it. So the water is composed of these water molecules, and you have a bazillion of these water molecules condensed together, and then you have liquid water, right? And you have what you're seeing on the pot. Well, if you transfer the chemical energy that was stored in the gas molecules and you convert it into thermal energy, what that basically is doing, what thermal energy is doing, is you're making this water molecule move around really, really quickly. Even if water looks perfectly still, you understand that there's water molecules that are moving around constantly inside this glass. Right? How do you know this? Well, if you have a glass of water and you drop, let's say, a drop of food coloring into it, what happens? That food coloring doesn't just stay there, it moves around and it starts spreading until the entire glass of water changes color, right? It does that without you having to stir it or shake it or anything, which means that there's water molecules inside that very still looking glass that's constantly moving around. The same thing for a pot of water. There's water molecules inside that pot that are constantly moving around. What you're doing... Hello, students. Welcome back. So today is the 10th class, and today what we're going to be talking about is photosynthesis. Now, we've been talking a lot about how cells survive, right? We've been talking about how cells eat food, right? Eat as in literally they absorb food materials. And we also learned the different methods in which they can break down the food and gain energy from the energy that was stored in the chemical bonds within these food molecules, right? And we've also learned how they harness the energy in the chemical bonds into a different form, which we call ATP. And ATP, why is that so important? Well, it's the source of energy that the cell can readily use. It's the easiest form of energy that the cell can use, and that's why it's better for us to convert the chemical energy stored inside the food into a form that we can readily use later on, which is called ATP, right? So these are all the things that we'll be talking about so far. Today, we're starting a new topic, and it's called photosynthesis. Have you heard of this word before? Photosynthesis. Obviously, yeah. Where have you heard it? Probably in like a 2nd grade science class. Probably you did some kind of project based on this, right? Photosynthesis has to do with what? Trees and leaves and light, right? And energy. We all know this, but now what we're going to do is we're going to learn about the specifics. We're going to try to look at this topic in a much more serious, in a much more scientific view, and try to understand what is actually going on, right? But before we do that, what do we have to do? We kind of have to refresh our brains. We have to remind ourselves about the different concepts that we're going to be talking about. First, I want us to review energy one more time, just so it's more clear in our heads. Second of all, I want us to look at chloroplast, which is an organelle that we covered previously in the eukaryotic organelles class. And thirdly, I want us to look back in the theory of endosymbiosis, which of course, I'm sure you all remember, right? Has to do with what? Which 2 organelles are related to the theory of endosymbiosis? Yes, and yes, the mitochondria and the chloroplast. Very good. So today, let's start off with energy. You've seen this before. Quickly review. Energy. There are many different types of energy in the universe. We all know this, right? What kind of types do we know? Well, there's thermal energy, there's chemical energy. What else? Electric energy, nuclear energy. What else? Kinetic energy, sure, and potential energy, right? All these different types of energies exist in the universe. Second point, energy cannot be created or destroyed, only transformed into different types. And I told you this was the first law of thermodynamics. Thermodynamics is a very, very difficult topic that we're probably going to cover in a physics class. So I'll see you then, right? But for now, we're studying biology, so we just know that energy cannot be created nor destroyed. It's kind of like, it's very philosophical, right? It's almost kind of religious in a sense. There is nothing created, nothing destroyed. Energy simply is, right? Energy simply is. But energy can exist. It can be transformed into different forms of energy, right? And the classic example was cell respiration, right? We're transforming chemical energy stored in the food molecules, and we're transforming it into what form? It's another type of chemical form, but it's now stored in the chemical bonds between phosphate molecules. Where? In the ATP. Am I making sense? Is it all clear in your head? It should be. If it's not clear, I want you to go back and look at cell respiration one more time, because it's really, really important to know cell respiration before you move on to photosynthesis. And we'll understand why in like 5 minutes, okay? So if you really don't understand what I'm talking about, go back. Look at cell respiration. If you do, good. Let's go on. Living organisms must be able to harness energy to fit their needs. This is what I've been talking about all along, right? Just because you're hungry doesn't mean that you can automatically stick your fingers inside like a wall socket and like electrocute yourself and be like, oh, I gained energy, now I can go on with my day. It doesn't happen, right? Why? Why is that not possible? Because I, as a living organism, I, as a human being, require energy. That is true. Why can't I gain energy from, the electric outlet on my wall? It's because the energy that's provided to me through that socket is electric energy. I don't need that particular type of energy. I don't need that form of energy. Instead, what I do need is ATP. Because my body requires ATP, which is a chemical form of energy. I can't use the electric form of energy. So what do I do to make ATP? I have to harness energy from somewhere else to make ATP. Where is that source? It's food, right? That's why we digest things. That's why we eat. It's because we gain energy from food, okay? Moving on. Another point, cells must use energy to sustain life. As I told you before, our cells are actually super, super busy. They do a lot of different things at once. What do? Well, they make proteins for one, yeah, right? Remember what organelles make proteins? Ribosomes, right? Ribosomes, and also the ribosomes that are attached to the rough endoplasmic reticulum, they all play a role in creating protein. What else? They're moving substances, right? So moving things within the cell as well as moving things out of the cell and both into the cell as well, right? Also, what is the cell doing? It's also dividing. These are all things that require energy. The cell must have a continuous supply of energy to make all these things happen. Another thing, cells gain energy by breaking down chemical bonds from food particles. It's just self-explanatory. I've already covered this, so let's move on. Cells store this energy as ATP for later use, of course, right? Remember, ATP is the most easiest source of energy that our body can use, right? So when we eat a lot of food, what do we do with the excess chemical bonds there? We all break it down and we make a lot of ATP. And what do we do with that? We store it in like a closet somewhere so we can take it out and use it later on when we need it, right? So this is the official technical chemical formula that explains cell respiration, right? I want you to look at this diagram very closely. I want you to start off with this. What is this? Look at this. This hexagon shape, what does this remind you of? Yes, glucose. How do you know it's glucose? There's 6 carbons, there's 12 hydrogens, and 6 oxygen atoms, 6 oxygen atoms put all together create one glucose molecule, right? And if you haven't taken chemistry yet, don't worry too much about it. Just look at the carbon atoms. There's 6 carbon atoms there, which is usually a good sign that it's a glucose, okay? This glucose meets what? It meets oxygen gas. And what happens when glucose meets oxygen gas? AKA, what happens when glucose is supplied to the cell in an aerobic environment, an environment that has a lot of oxygen in it. Well, we all learned this, right? First, carbon dioxide is produced. Second, water is produced. And third, the most important part is energy is produced, right? ATP is produced in this form. And this process occurs in 3 different steps. Remember this? Cell respiration, 3 different steps. First being what? Glycolysis. Where does it occur? Cytoplasm. Very good. Number 2, what's the method? Citric acid cycle. Very good. Number 3, the electron transport chain, aka oxidative phosphorylation. Where does that occur? The last two occurs inside the mitochondria. Very good. Remember the protein complexes, the 4 protein complexes stuck in the inner membrane of the mitochondria, right? And I told you about like the different gradients, the proton gradients. There's a high concentration of H plus on one side, low concentration of H plus on the other side. And this creates like a natural, a universal law from high to low gradients. You know what I'm talking about, right? So the cell produces ATP in this way. It uses glucose and it creates energy. It creates ATP. Let's move on. We're going to be talking about photosynthesis, right? That's the main topic for today. So let's kind of break down what photosynthesis means. What does it mean? Well, let's divide this word in half like this, right? The first part is photo, second part is synthesis. What is photo? If I ask you, what is a photo? You'll probably, look at me like I'm a dumbass and be like, a photo is a photograph, right? True, technically, but what is a photograph? Explain that word to me. Well, graph, like you heard in probably the first class, right? I talked about biography and autobiographies. What are those? Those are basically books depicting what? A person's life, right? Bio meaning life. Graphy means to depict something, to draw something, right? So computer graphics, again, it's drawings created by a computer. So a photograph in this instance would be it's drawings produced by light, which is exactly what a picture is, what a photograph is, right? A camera lens captures light and it creates a picture. That is a photograph. Good. So a photo in this sense means what? It means light. Okay? It means light. What is synthesis then? When you synthesize something, what does that mean? Synthesize means to make something or to create something. Okay? For those of you who are musicians, you probably know what I'm talking about. What is a synthesizer? Do you know what that is? It's actually a musical instrument, right? And what do? It looks like a keyboard. What do you do with it? It produces a lot of different sounds that technically you can't make. Like, for example, if you're like, I don't know, you're like Skrillex Jr., right? And you want to create the latest, fire beat, right? So you're like slamming down on the keyboards and you're creating all these weird mechanical noises. You're not actually doing that on a piano, right? You're pressing keys like it looks like a piano, but it's not a piano. What is it? It's a synthesizer. It's a keyboard that can create a lot of different sounds. So synthesize in this sense means to create. Hello, students. It is I, your online biology teacher, and today we're going to be talking about DNA, genes, and chromosomes. Like previous chapters, there's a lot of information stored in this concept, okay? So I decided it's best to divide this into 2 separate parts, and this is going to be part 1. So what are we going to be talking about today? We're going to be talking about DNA structures, right? We talked about DNA structures before, so hopefully this will mostly be a review, but just in case you were a little unsure or you just kind of breezed past it, we'll look at the DNA structure extensively, try to figure out what's actually going on, and then we'll also be talking about proteins. Proteins, super, super important. Very, very important proteins. And now you might be looking at me like, what isn't important? Every single thing that you say is like really important. I should always remember it. Okay? I concede, I agree, but proteins really are super, super important. I mean, I cannot emphasize this enough. Proteins are very, very important, and you'll see why. You'll see why in just a bit, okay? So bear with me for a second. So, first things first, let's talk about DNA structure. Deoxyribose nucleic acid, DNA, is composed of 3 major sections, and you're looking at me like, yeah, I already know this. Good for you, right? 3 major sections. What were they? Do you remember the names? What was this? This pentagon shape. This was called deoxyribose, right? And I told you, don't worry too much about what it is. Just imagine it to be like a friend of glucose, right? Glucose had 6 carbons. This deoxyribose has 5 carbons. So it has a pentagon shape, not a hexagon. I am tripping up on these 2 words today. I don't know why. But anyways, so it's a pentagon shape. Next to that is a phosphate group. What is a phosphate group? Remember, it's a phosphorus atom with 4 oxygen atoms attached to it, right? So this big P with the circle around it, this is a phosphate group. Next to that, attached to the opposite side of the deoxyribose is a nitrogenous base. This part over here. right? I feel like we've talked about these two things a lot, but we haven't really covered the nitrogenous base part, right? So we'll talk about what these bases are. Nitrogenous base. What is it? The nitrogenous base in DNA comes in 4 different types. Adenine, thymine, cytosine, and guanine. You should remember those 4. You have to remember those 4. And you're looking at me like those are totally weird words that I've never even seen before. Okay, to make it easier for you, let's call adenine A, thymine T, cytosine C, guanine and G. We take the first letter of the word and we represent it like that, right? A, T, C, G. That's all you have to remember. There's 4 types of nitrogenous bases. So where are they? Let's look at the structure right here, okay? Pay attention to this part. What is this part? It's a ribose. Look at the blue parts, right? It's a ribose. Next to it, the yellow circles are phosphate groups, right? And as we know, the ribose, the deoxyribose, and the phosphate groups connect and form one big chain, which acts as the backbone. Remember the 2 strings in a double helix form, right? It's one of the strings. That string itself is made up of phosphorus and sugar, right? So we have the sugar phosphate backbone, but attached to the deoxyribose is another thing. It's the nitrogenous bases, right? So you have 4 types over here. Here's what you should do, okay? Squint your eyes, and then kind of move away from the monitor. Opposite of what I usually tell you to, like, don't put your nose on the monitor, kind of move away from it, and squint at this, okay? You're looking at the vague shape here. What does this vaguely look like? It looks like one big circle, yes? What does this look like? Like lean back, like totally lean back, okay? What does this look like? 2 circles. Are you with me so far? What does this look like? 1 circle. What does this look like? 2 circles, okay? So what we can say is that the nitrogenous bases, there's 2 types. One type is one big circle. The second type is one big circle and one small circle together, okay? So there's 2 different types of nitrogenous bases. They have names. Their names are A, T, C, or G. This is a T, this is an A, this is a C, and this is a G, okay? Here's what you have to remember about the nitrogenous bases, okay? You have to know the names. If you don't know the names, at least the first letters of each word, and you have to kind of know where they're located, right? You have to know that there's a deoxyribose phosphate group backbone, and then there's nitrogenous bases that's extending out this way. What does that mean? Well, let me show you. This is a DNA, right? This is a DNA backbone. This is made out of phosphate and sugar, and these are nitrogenous bases that are sticking out like this. Are you with me so far? Right? Here's another string. The thing about this string is that it's flipped over. Remember, double helix is 2 strings, and they have opposite. They're anti-parallel to each other, right? Like the opposite side, this also has nitrogenous bases sticking out like this. Are you with me so far? Okay? If you take these 2 strings and you hold on to the top and you twirl the bottom, what do you end up with? You end up with the double helix form, okay? So what are these nitrogenous bases? Well, some of them are one large circle, some of them are one small circle and one large circle. Are you following me so far? Right? And these names are A, T, or C, or G. Likewise, in the opposite string, they have nitrogenous bases. Some of them have 2 circles, some of them have 1 circle. Like the opposite side, they also have the names. The nitrogenous bases also have names, and they're called A, T, and C, and G. Okay? Here's what you have to know. You have to know the relationship between the nitrogenous bases. Okay? You have to know who's friends with who. That's all you have to know. Who's friends with who? T, thymine, is friends with adenine, A. T and A, BFFs. Okay? What does that mean? They're best friends. They don't hang out with anybody else. Anybody else meaning C or G. T doesn't hang out with C or G. It only hangs out with A. A only hangs out with T. Doesn't hang out with C or G. Do you get it? C and G, obviously, BFFs, right? What does that mean? C and only hangs out with G. G only hangs out with C. It's an exclusive relationship. Okay, so you have to know the relationship between these nitrogenous bases. And that's it. Let's talk about the nitrogenous bases a little bit more. So, the DNA structure. The sugar phosphate backbone connects together to form one long strand. Okay, so deoxyribose connected with the phosphate group connects with another deoxyribose connects with another phosphate group. It forms one long continuous chain like this. Okay. Two of these strands running opposite directions from each other connect and form a double helix. I told you before, if you have the top part of the string right here, bottom part of the string right here, and you have another string, you have to flip it over. So this is technically the top part. Okay? This is the top part, that's the bottom part, and they're running anti-parallel to each other. Okay? And when you twist these two strings around, if you intertwine these two strings, they form the double helix. Let's look at this photograph. This is showing you the actual structure of the DNA. I'm going to start with the, I'm going to start with the middle picture right here, okay? This is the middle picture. Okay? Look at this. What is this? What is the blue part? Deoxyribose. What's the yellow circles? Phosphate groups, okay? So these are forming the phosphate sugar backbone. Take a look at the second string though. Look at the ribosome. Look at the ribose. Look at the ribose. See how it's actually flipped over this way, flipped over 180 degrees. So this is actually running in the opposite direction, while this is running like this. Do you see it? So these strings are anti-parallel to each other. Notice the nitrogenous bases right here. Right here, right? Some of them have one circle, some of them have two circles, and they're connected to the opposite string's nitrogenous base. Okay? In this case, we have T over here, and I told you T only hangs out with who? With A. So T only holds hands with A. Down here, you have C, right? If you have C here, C only hangs out with who? G, right? So G from the opposite string holds hands with the C from this part of the string. Are you following me so far? And these two nitrogenous bases hold hand like this. If you look at this structure, notice the blue deoxyribose and the yellow phosphate groups, the blue deoxyribose and the yellow phosphate groups. So they form this backbone structure right here. Okay? They form this backbone structure. The rungs inside the ladders like this are essentially can be divided in half. So one nitrogenous base is holding on to the hands of another nitrogenous base of the opposite strand. Okay? And this is an actual... Hello students. Welcome to the 18th lecture. What are we going to be talking about today? We're going to be talking about Mendelian genetics. Now, this is the first part of a really long series in which we cover the basics of genetics. Genetics is one of those things where you have to get it right the first time, or else you get it wrong all the time. And what I mean by that is genetics is a field in biology that it comes up again and again as you progress through your school years, right? So if you're learning it right now, let's say in 7th, 8th, or 9th grade, this will come up again in junior year, maybe when you're taking AP biology. This will come up again in your college years when you're taking introductory biology in the college level, okay? And the thing is, it's not that different. Remember when I talked about mathematics, when I said, you know, you have really simple algebra in the very beginning, and then you move on to like pre-cal, and then you get to like multivariable calculus, and these are progressively more difficult, right? Genetics, not so much. As you progress up, you will find that it's actually very, very similar. There's not a lot of things that are added to this. So it's very important for you to get this right the first time. And if you get it right the first time, the next 6 years or so of biology is going to be a breeze for you because everything is just repeated one more time. Okay, so today we're going to be talking about Mendelian genetics. Specifically, we're going to be talking about who is Grigor Mendel. You've heard of this man before, right? And you've probably heard it, passing by kind of manner, right? Your teacher, referred to him once, like for a minute or so in the classroom and then just moved on. I don't want to do that. I want to spend a little bit more time talking about who Grigor Mendel is because he was a very influential figure. And because his life is very interesting, and hopefully I'll be able to persuade you that his life was very interesting indeed. Secondly, we'll be talking about the key vocabulary words. So a lot of students struggle with this area of genetics, and the reason why they struggle is because they're not clear about what these individual words mean. And what I mean by that is there's going to be some key words that we're going to be covering in the later part of this lecture. And you have to get these definitions down pat. For example, if I tell you, think of a cloud, you don't spend like 2 seconds thinking about what cloud means, right? It just immediately pops up into your head. That's the state you have to be with these terminologies in genetics. When I mention the words like homozygous recessive, you can't spend like a second or so thinking about what that word means. You have to just immediately know what it means. And to get to that level of immediately recognizing the word, you have to repeat it over and over again so that you don't forget the definitions, right? So we're going to be covering the key vocabularies for genetics. So first things first, who is Gregor Mendel? I want to tell you a story. Okay, so put your pens down. Okay, this is story time, children, so gather around. I'm going to be telling you about the life of Gregor Mendel. Here he is. What does he look like? He kind of looks like Harry Potter, right? He has like the whole really frowny expression. He has very small glasses on. He has like, the typical male pattern baldness going on. He has this weird like bling bling necklace. This is not a gangster sign or anything, okay? This is actually, he was a Catholic priest, okay? So Gregor Mendel, he actually lived in Czech. He didn't live in the country Czech. He lived in the region that later became Czech, and he was a priest. And he lived between the years of 1822 to 1884. Gregor Mendel is often referred to as the father of genetics, and I will tell you why, okay? So Gregor Mendel, he was actually the son of a farmer. His entire family was actually a farmer family. So his dad and his dad's dad and his dad's dad's dad were all farmers. They've been farmers all along. And Mendel was born into that family. And when he was around the age of like 12 or 13, he went to his dad and he says this. He says, "Dad," and his dad says, "Yes, son." And he says, "Dad, I have to tell you something, okay? And no, I'm not gay. Don't worry. Okay, I have to tell you something. I don't want to be a farmer. I don't want to be a farmer. I think that my future lies in studies. I want to go to college." Very different from you guys, right? He's very serious about his studies, and he was very, very smart. He was a very intelligent child. And he went to his dad and he said, "I don't want to be a farmer." And Mendel's dad said this. He says, "Son, it's true. You are very smart, you are very intelligent, and I also believe that you shouldn't be a farmer, you shouldn't waste your potential. However, if you want to study, you have to go to a university, and because we are farmers, because we are a poor family, we can't afford to send you to college." That's really cool of Mendel's dad, by the way, right? This is 1820, 1830s, okay? Imagine yourself as a farmer in the 1830s and your son comes up to you and he says, he has this weird attitude and he's wearing like skinny jeans and he comes up to you and he's like, "Dad, your job sucks, man. I don't want to be a farmer." What do you say? Immediately you just, slap in the back of the head. You're like, "Get back to work, you slave," right? No, Mendel's dad was awesome. He was really cool. And he said, "Mendel, I believe you. I believe that your future lies in studies and I don't think you should be a farmer, but I can't afford to send you to college." So Mendel, upon hearing this news, becomes very depressed and he says, "Okay, I guess my fate is sealed. I guess I have to be a farmer." So he's like very depressed. But a couple days later, he hears about a monastery near his house. And what is a monastery? It's basically a place where people come and study religion. People who devote their entire lives to studying theology, to studying religion, live together in a monastery. And he says, maybe if I go into that monastery and become a monk, and remember, monk is not a Buddhist monk here. I'm talking about, people who study religion for a living, okay? If I become a monk, then maybe I can devote my time into studies without having to, work in the field. So he goes to the monastery, okay? He goes to the monastery and says, "I want to study theology and I want to live in this monastery," right? And there's a person that's running the monastery, right? So the president of the monastery, probably some head priest, comes up and he says, "Hey, I heard you're Gregor Mendel." And Mendel says, "Yes, that is me." And the head priest says, "I heard you're pretty smart." And Mendel says, I'm not like brilliant, but I'm okay, I guess. And the head priest says this, okay? I'll make a deal with you. Here's the deal. I will clothe you, I will feed you, and I will give you a place to sleep, okay? So you can live in the monastery, but you do something for me. And Mendel says, what? I'll do anything if you just give me time to study. So the head priest says this, okay? I want you to do something for me. I want you to try to answer this question. And here's the question. What is God's will? Yes, exactly. Right? That's the response that Mendel had. Mendel was like, what is God's will? How do I know? Right? But he has to do it, right? He has to make this deal happen in order for him to continue studying. So he goes into the monastery, he gets accepted, and he has this question laid upon him. So he spends weeks just frustrated thinking about how he can answer this question. And I'm going to ask you the same question. What is God's will? How do you test that? How do you know that? Mendel writes in his diary, he says, it's been week 2 of living in this monastery. I've been trying to find out ways to answer this question. I got nothing. So he spends his time walking around the monastery and he starts making this garden. And he starts caring for this garden. Remember, he's the son of a farmer, so he's very used to taking care of plants. So he creates a garden for himself and he looks at the peas that he's growing inside of his garden. And he notices something very peculiar about the peas. And for those of you who are familiar with Mendel's story, you know where I'm going with this, okay? So he looks at these peas and he says, something's weird about these peas. Some of the peas are green and some of the peas are yellow. I wonder what will happen if I just start randomly mixing them together. So he takes the plants that produce green peas, he takes the plant that produces yellow peas, and he starts crossbreeding them together and seeing what kind of peas arise. And he does this thousands of times. Okay? What he's doing is he's gathering all the new peas that are produced. He's looking at them by the candlelight. Remember, this is 18-something, right? 1840s. Right? He's looking at these peas and he's basically categorizing them based on different traits. The trait being the color. So he says, this is a yellow pea and this is a green pea. And he does this experiment millions and millions of times. And he gathers this huge amount of data. And this is what he noticed. There's about 740 yellow peas. And there's about 282 green peas. Yes, exactly, right? These numbers don't mean anything to you, right? But remember, Gregor Mendel was a very, very smart person. So he looked at these numbers and he says, something's fishy about these numbers. Let me try the experiment one more time. He does the same experiment one more time. He gathers the data, he looks at the numbers and he says, yellow peas, how many are there? Well, there's about 745.